Human episodic memory does not operate like a video camera passively recording a timeline of events. It functions as a highly selective network, constantly prioritizing certain data points while deliberately severing connections to others. For decades, both the public and professionals misunderstood how emotion influences this network. The prevailing assumption was a concept called emotion-enhanced memory, or EEM. This theory treated emotion like a global volume dial, suggesting that a high-stress or highly positive situation simply cranks up the retention of an entire event. But that assumption contains a critical logical flaw. If physiological arousal uniformly amplified memory, we would possess perfect, comprehensive recall of our most traumatic or thrilling moments. Every detail of a car crash or a wedding day would be crystalline. In reality, memory assessments of public disasters and personal triumphs show the exact opposite. Highly effective memories feature profound gaps, reveal severe distortions, and the complete omission of facts. Effective arousal functions as an aggressive, localized filter that physically reconfigures the architecture of the memory trace. This selective filtering creates what researchers call a memory trade-off. Highly prioritized core details are burned into the neural network, but that exact prioritization occurs at the direct expense of the peripheral context. You can observe this directly in the weapon focus effect. A victim of an armed robbery will often have flawless, vivid recall of the barrel of the gun, but fail entirely when asked to describe the dimensions of the room or the face of the perpetrator holding it. This leaves a biological question. In the fraction of a second an event occurs, how does the brain mechanically decide which specific details survive the filter and which are discarded? The sorting happens through neurochemical tagging. The brain releases specific compounds that effectively tag high-priority stimuli, creating localized neural hotspots that guarantee those details are encoded. To understand why our most intense memories are so fragmented, we have to deconstruct the exact mechanisms of how these tags are encoded, and later, how they are updated. Proving that emotion chemically alters memory presents a difficult experimental problem. Emotional stimuli, like an image of a snake, naturally capture our attention more than neutral stimuli, like an image of a bookshelf. We have to separate the simple act of paying attention from a genuine biological memory enhancement. When tested immediately, recall rates for emotional and neutral stimuli are identical, driven by attention. But 24 hours later, neutral data degrades sharply, while emotional data remains elevated. Structure also dictates retention. When lists are intermixed, emotional data actively cannibalizes the neutral. Grouping emotional stimuli together makes them highly distinct, and that distinctiveness artificially mimics memory enhancement through simple novelty. By utilizing intermixed lists and delaying the recall test by hours or days, researchers strip away the cognitive noise of attention. What remains is the isolated biological effect of emotional memory encoding. True emotion-enhanced memory is not a byproduct of simply noticing something. It is a distinct, time-dependent neurochemical process that requires a physical delay to consolidate. Knowing that this enhancement is biological brings us to the mechanics of the trade-off. How does the brain actively tag a core detail while simultaneously suppressing the background? The answer lies in the GAIN model, which stands for Glutamate Amplifies Noradrenergic Effects. High physiological arousal immediately triggers diffuse inhibition, bathing the neural network and causing baseline activity to flatline. But in isolated areas already experiencing high threat activity, beta-adrenergic receptors ignite. These trigger targeted explosions of glutamate, creating extreme localized spikes of excitation. The entire surrounding neural network is subjected to widespread suppression, leaving only these hyper-localized bursts of chemical excitation. This neurochemical contrast is the precise mechanism behind the weapon focus effect. The gun gets the glutamate burst, the perpetrator's face gets the diffuse inhibition. The GAIN model reveals that the brain actively suppresses background information to create the hyperfocus needed to amplify a single point. Scaling up from cellular chemistry to broader brain networks, this tagging process is heavily controlled by the amygdala. Researchers Kark and Kensinger tracked this control center using fMRI scanning, measuring objective physical arousal, specifically heart rate deceleration, while subjects encoded new memories. Their scans isolated a clear pattern. A spike in physical arousal drastically increases connectivity between the amygdala and visuosensory regions. The strength of this exact connection directly predicts how vivid the memory will be. But the data contained a highly specific exception. This amygdala-driven tagging only correlated with memory vividness for negative stimuli. The connection did not exist for positive or neutral events. The brain's affective tagging architecture is strictly biased. It prioritizes threats over rewards, trading a balanced view of reality for a survival advantage. This survival architecture is so aggressive that it actually manipulates memory consolidation timelines. The tagging process doesn't stop when an event ends. This timeline diagrams a paradigm by Joseph Dunsmore. At T0, neutral icons of tools and animals register equally as low-priority memories. Minutes later, the subjects see new images from those categories, but the animal category is paired with a mild shock. 24 hours later, participants are tested on their retention of the neutral images seen before any shocks. The results showed the original, unshocked animal items were remembered significantly better. A retroactive signal upgraded their priority status. Downstream emotional learning acts as a retroactive signal. The brain realizes information is dangerous, reaches back into recent storage, and rescues structurally related memories. Episodic memory traces remain highly malleable for a specific window. This allows subsequent threats to retroactively dictate exactly what we remember about the past. This creates a final paradox in the memory network. If affective tagging makes a trace incredibly durable and capable of retroactive enhancement, does that trace remain flexible enough to accept new, non-threatening context? This 2x2 matrix plots the findings of memory updating research by Mather and Knight. Neutral memories fall in the bottom right. They have moderate retention strength but high flexibility, meaning they are easy to update. In their study, participants were asked to learn new spatial locations for faces they had previously associated with either neutral imagery or highly negative emotional imagery. The participants failed to behaviorally adapt, struggling to integrate new location data for faces with an affective tag. The intense neurochemical prioritization required to cement an emotional memory heavily impairs the brain's ability to append new associations to that trace later on. The exact mechanism that guarantees an emotional memory's long-term survival simultaneously strips it of its ability to be updated. Affective memory operates strictly on a dual nature. The core details of an emotional event are exceptionally durable, but the memory trace itself becomes intensely rigid. Neutral memories lack that extreme durability, but they remain agile, easily integrating new contextual information as our environment changes. This massive disparity is not a psychological accident. It is explicitly engineered by the localized glutamate bursts of the GANE model and the survival bias of the amygdala. The human brain actively sacrifices precise historical accuracy. It discards background context and rejects new updates in order to lock in high-contrast survival parameters. The affective system renders memory aggressively prioritized, permanently trading its capacity for flexible updates for the sake of survival.